The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hi children, I will begin the session with a quote. Make all your best effort with vigor and determination. Face all problems with self-confidence. Act only thoughtfully. Never indulge in any mindless activities. Swami Nirmalanand Yogi. So my dear children, again I am back with you to join this virtual class. Here we are going to recap the third chapter, and for the recap I welcome our 3 stars of Gurukulam school, and these 3 stars are Sachin, Shobit and Alina. So we all welcome these 3 children for the session.
Hello everyone, I am here to give a quick recap on our previous classes of botany. In the previous class, we learned about animal husbandry. Now, what is animal husbandry? The agricultural practice of breeding and rearing livestock is called animal husbandry. It deals with cows, buffaloes, sheep, horses, etc. In the dairy farm management, it is the management of animals for milk and its products. Important points in dairy farm management is it should be well housed. That is, the house should be in a proper location and should be with the proper facilities. Feeding should be made in scientific manner. Quantity and quality of fodder should be maintained. Cleanliness and hygiene should be maintained to get a disease-free varieties. Adequate water should be provided. Regular visit by a veterinary doctor is mandatory. Poultry farm management. It is the management of animals for eggs and meat. Selection of suitable breed. Proper safe conditions should be maintained. Proper feed and water should be provided. Hygiene and health should also be maintained. Animal breeding. A group of animals related by descent and similar in most characters like appearance, features, sizes, etc. are said to be a breed. There are 2 types of breeding, that is inbreeding and outbreeding. Now, what is inbreeding? Inbreeding is mating of closely related individuals within the same breed for 4 to 6 generations. Superior male and female are selected from the same breed and are done to be mated. More milk per lactation is the criteria for superior female. For cows and buffaloes, superior male gives rise to superior progeny. Progeny is the offspring. Increases homozygosity, that is, same alleles or genes are carried from the parents to the offspring. Helps in accumulation of superior genes. Continuous inbreeding reduces fertility and even productivity. This is called inbreeding depression. Outbreeding. It is the breeding of unrelated animals, that is, of 2 different breeds. Outcrossing. Mating of animals of same breed but have no common offsprings. No common ancestors up to 4 to 6 generations is called outcrossing. Offspring of such mating is called outcross. A single outcross helps to improve inbreeding depression. Crossbreeding. Mating of superior males and females of different breeds. It allows desirable qualities of 2 different breeds to be combined. Interspecific hybridization. Mating of 2 different species of animals is called interspecific hybridization. Artificial insemination. Controlled breeding experiments are carried out using artificial insemination. The semen is collected from the male and is injected into the female reproductive tract of the selected female. The semen collected may be frozen and can be used for later. For the later purposes, it can be stored. Then semen can be transported to where the female is housed in a frozen form. These are the advantages of artificial insemination. Then we learned about multiple ovulation embryo transfer technology, that is MOIT. It is used to improve chances of successful production of hybrids. Cow is administered with hormones like FSH, that is, it can induce follicular maturation and also superovulation. If a cow is supposed to produce a single egg per cycle, but after FSH is induced into the cow, it produces 6 to 8 eggs. Then the female is either mated with an elite bull, that is a superior bull, or artificially inseminated. Then the fertilized egg that develops to form to a 8 to 32 cell stage is later taken and is given singly to surrogate mothers. That is, each egg is provided to other surrogate mother cows. Then the genetic mother is available for another round of superovulation. This technology is used to increase herd size in a short time. Then we learned about beekeeping. Beekeeping is also called apiculture. It includes maintenance of hives for honeybees for the production of honey. Honey is a food of high nutritive value and also is used as medicines. Honeybee also produces bee wax, that is useful for industrial purposes, like for the production of cosmetics and also used in polishes. Apis indica is most common species used in beehive or used in apiculture. For successful beekeeping, one must know the following points. Knowledge of the nature and habits of bees, selection of suitable location for keeping the bees, catching and hiving of swarms, management of beehives during different seasons, handling and collection of honey and bee wax. Bees are the pollinators for many plants. So if we keep beehives near crops during the flowering season, there will be increased pollination and also increased honey yield. Fishery industry is related to catching, processing and selling of fish, of either aquatic or also marine fishes. Common freshwater fishes are cutla and rocco. Common marine fishes are hilsa and sardines. Production of aquatic plants and animals, both freshwater and marine, is called by pisciculture and aquaculture respectively. Increasing production of the fish is called blue revolution. Thank you.
Till this we have learned about animal breeding. From here we learned about plant breeding. Plant breeding is the purposeful manipulation of the different plant species for the development of desirable plant type for improved cultivation, yield and disease resistance. What are the objectives of plant breeding? Improve yield, improve nutritional quality, develop increased tolerance against the environmental stress and insect pests, develop increased resistance against the pathogens. The next topic that we had studied was main steps for the development of a new varieties through plant breeding. There are 5 steps and the first step is collection of variability. The collection and the preservation of different wild varieties, species and relatives of cultivated species. In that topic we have learned a new term called germplasm collection. The entire collection of all the diverse alleles for all genes for a crop is termed as germplasm collection. The second step is evaluation and selection of the parent. All the entire germplasm are evaluated for the identifications of the plant with desirable characters. Out of the selected plants, one is marked as female and the other are marked as male. Then the third step is cross hybridization among the parents. The cross hybridization between the parents lead to the formation of the hybrid with desired characters. And the fourth step is selection and the testing of superior recombinants. In this, it involves the selection of desired plant type among the progeny of the hybrids with the help of scientific evaluations. The selected plant is superior to both the parents. The superior hybrid is then self-pollinated for further generation. And the fifth step is testing, release and commercialization of the new variety. The new variety that had been developed are tested for its duration, quality, effect of weather, yield, disease and insect pest resistance. Then we learned about the crop and its new variety. Wheat has a new variety called Sonalica and Kalyansona, while the rice has a variety called IRA, which is seen in Philippines, Jaya and Ratna, which is developed in India. In the case of sugarcane, Saccharum barbarii crosses with the Saccharum officinarum for the formation of a new variety which has high yield, high sugar content and thick stem. The next topic that we had studied was the plant breeding for the development of disease-resistant plant. The objective of this is to enhance the food production, reduce the consumption of the chemicals, bactericides and fungicides. Later, we discussed about the pathogens and the disease caused by it. The fungus causes the disease such as brown rust on the wheat, red rot on sugarcane and late blight in potato. Bacteria causes disease such as black rot in crucifers, and the virus causes the disease like tobacco mosaic and turnip mosaic. There are two methods of breeding through which a disease-resistant plant can be created. The first one is conventional breeding and the second one is mutational breeding. The disadvantages of the conventional breeding is that the availability of all genes is limited. Then we further learned about the crops and the new variety that had been developed with the help of conventional breeding. The first crop is wheat, which had a new variety called Himagiri and is resistant to the disease called leaf and stripe crust Hilbert. The second crop is Brassica and its variety name is Pusaswarni and is resistant to the disease called white rust. And the third crop is cauliflower and its variety name is Pusa Shubra and Pusa Snowball K1 and is resistant to the disease black rot and curled blight black rot. And the fourth crop is cowpea and its variety name is Pusa Komal, which is resistant to the disease bacterial blight. And the fifth crop is chili and its variety name is Pusa Sadabahar and resistant towards chili mosaic virus, tobacco mosaic virus and leaf curl. The next topic that we had studied was mutation breeding. Mutation breeding is the process by which the genetic variation takes place with the change in the base sequence within the gene, which lead for the formation of a new character. How we can do a mutation breeding? Induced mutation artificially by chemicals. And the second point is selecting and using the plant of desired characters and screening of that plant for disease resistance. Next topic that we studied was the plant breeding for developing the resistance to the insect pest. And the first one is hairy leafing plant which resists the insect pest. And the second one is solid stem in weed which resists the stems of life, and the third one is smooth leaves, nectarless cotton varieties which resist the attack of bollworms, and the fourth one is high aspartic acid, low nitrogen and sugar content in maize which resist the maize stem borer. And we discussed about different crops and its variety which are resistant to certain insect pests. The new varieties such as Pusa Gaurav of the crop Brassica is resistant towards the aphid, and varieties such as Pusa Sem2 and Pusa Sem3 of the crop Flatbean is resistant towards jassids, aphids and fruit borer. And the third variety, that is Pusa Savani and Pusa A4 of the crop Okra is resistant towards the shoot or fruit borer. Then we studied about the plant breeding for improved food quality. In this topic, we learned about a term called hidden hunger. The symptom which shows the deficiency of minerals, nutrients and vitamins is termed as hidden hunger. Then we discussed about the topic biofortifications. Biofortification is the method of breeding the crop with increased nutritional quality like vitamins, nutrients and higher proteins and healthier fat in a crop. The objectives of the biofortification is improving the protein content and quality, improving the oil content and quality, improving the vitamin content, improving the micronutrients and minerals.
So let's first go through the single cell protein. Single cell protein is a microbial biomass which is used as food or feed. It is an alternative protein source for animal and human from certain beneficial microorganisms like spirulina. Microbes like spirulina, Methylotrophus, Methylophilus can be grown in large industrial scale as source of good protein. Advantages of single cell protein are: it is easy to grow, nutrient rich, high yield and reduces environmental pollution. Now the next topic is tissue culture. Tissue culture is an in vitro technique of regeneration of whole plant from a part of a plant by growing it on a culture medium is called as tissue, under sterile condition is called as tissue culture. The part of the plant taken for tissue culture is called as explant, and the ability of explant or cell to grow completely into a whole plant is called as totipotency. Using this method, thousands of plants can be grown through tissue culture. This is called as micropropagation. Now, what are the advantages of tissue culture? Its seedless plants can be multiplied. A large number of plants can be produced in a short interval of time. Plants in which sexual reproduction are absent can undergo somatic hybridization. Now, what is somatic hybridization? The fusion of protoplast of somatic cells of different varieties of plants from different varieties of species in a suitable nutrient medium to obtain, that is, the suitable nutrient medium is in vitro, to obtain a somatic hybrid is called as somatic hybridization. There are certain methods to do this. The steps are as follows. First, the single cells from selected plants are isolated. The cell walls of the plants are digested by using enzymes like pectinase and cellulase to expose the naked part, naked protoplast. The naked protoplast surrounded by plasma membrane is isolated. The protoplast which is isolated is fused with the hybrid protoplast to produce a somatic hybrid plant. The somatic hybrid plant, for example, can be given as pomato.
I thank Sachin, Shobit and Alina for taking such a beautiful session related to strategies in the enhancement of food production. So my dear children, with this, the third chapter, I think you will be able to understand very nicely, as these children, they have made you more clearly all the concept. And with this, I wind up the session and eagerly waiting to join with you in the next session. Have a great day.